Okay, so we covered all the parts of the cell, both prokaryotic, eukaryotic, and protozoan. Now let's take a look at what happens when we want to make copies of the cell. Now, if you're talking about a single-celled organism like bacteria, its processes are a little simple, but we're going to try to focus on eukaryotes. And when we're talking about DNA replications, one of probably the most important and complicated of the copying portions of the cell, we have to consider how important it is to make sure that the message stays the same from cell to cell. So think about it. You're made of 1,000,000,000,000 cells or so. All trillion cells have to have the same DNA information, although different parts of it are going to be turned on and off depending on what tissue it belongs in. You don't want the same genes turned on in your eye, for instance, that you have in your heart. Otherwise, you'd have a bunch of eyeballs for a heart, and that just wouldn't be any good. So what you need is you need to be able to first, before we talk about how we turn these genes on and off and what are genes, let's talk about how we copy it. And before we start talking about how we copy it, let's think about what the different parts of the cell cycle are. The cells have different life stages, and so let's consider what those stages might be and how important they are to a particular life or cell. So the first portion, and you can start anywhere really. It's a circle, so it really doesn't really matter where you start. But if you consider that you have the mitosis phase, let's think about it right after the cell has divided. You have a brand new cell right here. This is a birth of a new cell, 2 new cells, right? Because mitosis will give you 2 new cells. So you have these 2 new cells. It has this one phase called G1 or gap phase. This is in everything other than M phase is considered interphase. So interphase is considered everything in between cell divisions. And because one of the big purposes of life, one of the big characteristics of life is that we're able to copy ourselves. We're able to make copies of ourselves and inherit information. Mitosis or cell division is an important phase of life. However, there are other things that our cells have to do. Our glands have to release hormones. Our heart muscles have to contract. Our lung cells have to absorb and oxygen and put out carbon dioxide. All these things have to continuously happen, and so the day-to-day functions of the cell happen in here, in between the cell divisions. So cells divide. Obviously, early on when you were in your mother's womb, you were going through a lot of these cell cycles. And as you get older, you go through less of these cell cycles. If you're a skin cell, then you have to do this a lot, right? Because you're constantly doing mitosis. You're constantly doing cell division so that you can replace dead or dying skin. The same in the lining of your mouth and your intestines. These cells receive a lot of wear and tear and have to replace themselves continuously. Blood cells as well. Red blood cells last only 3 months. And so as they age, they actually lose their nucleus. And so that they can function in their old age. They're not able to go through mitosis anymore. Their nucleus is gone, and they're just simply doing this. And they go into something called senescence. And senescence is a word that just means that the cell is ready to end. The cell will end rather than keep on replicating. There are these cell types called stem cells, blood stem cells in your bone marrow that are continuously replacing these dead or dying red blood cells. And we'll talk more about this when we talk about sickle cell a little deeper as we get into genetics. However, here in this section, in this phase of your cell cycle, the life of your cell, G1, the phase is where the cell is growing, it's making proteins, it's doing its day-to-day function. And at some point, the cell has a decision to make. And it has a decision whether to stay at G0, and G0 means the cell is paused. It's going to be doing its work. It's not going to be growing anymore. If the cell is going to continue to do mitosis, if it needs to make more copies of itself, and how would it know that? it's going to get signals from its neighbors. So let's say you stab yourself as you're working with a nail. Your cells, the neighboring cells, get the signal that there's damage, and those signals tell the cell it's time to do mitosis. We need to repair the broken tissue. So it leaves G0 and then enters again into mitosis. And during G1, what it does is it makes all the proteins that it's necessary for DNA replication, for the copying of DNA, all the enzymes that are necessary. It's making all the nucleotides that are going to be necessary to make a copy of your DNA. In S phase, that's this phase here, is also known as DNA synthesis phase. This is where DNA synthesis is going to happen. We call it, we're talking about it as DNA replication, but DNA replication is in fact synthesis, right? You're making a copy of the DNA. And there's three little phases of, the three phases of DNA synthesis, or S phase. There's the early, the mid, and then the late S phase. And in the S phase, all you're doing is making a copy of all your DNA. And that's a big deal, and it takes a while, and it takes a lot of effort and a lot of energy, a lot of ATP, and a lot of resources. So this is also, this gap, this G1 is, or gap 1 is where the cell is preparing for that DNA synthesis. If that cell did not have to do that, if it's a pancreatic cell whose job is to make insulin or glucagon, or if it's a nerve cell, nerve cells are famous for going into this phase where they're no longer ready to do mitosis. They're just going to do their job. They go into this phase called G0. So let's make sure we understand that there are checkpoints here that stop the cell from continuing on into doing any more of its growth or preparation for this mitotic phase. And this mitotic phase is where the cell does replication. These checkpoints are important, and in fact, mutations in these control points have been linked to a number of tumors and causes of cancer. So one of these checkpoints is here near the end of G1, and about here at this stage, there's a checkpoint that checks for DNA damage. All right, and if there's any DNA damage, then the cell does arrests in the cell cycle and doesn't go anywhere until the DNA is repaired or the cell goes into cell death. Then S phase starts, and there's a checkpoint to see if there's any unreplicated DNA in S phase. If there's DNA that hasn't been replicated near the end during S phase, then the cell cycle arrests or stops. It's called S arrest. I know it's kind of silly, but S arrest, right? So you have G1 arrest, where the cell stops its cell cycle here because of damage in the DNA that the cell recognizes. There's the S arrest, where the cell hasn't been able to replicate enough DNA, and theoretically it'll either complete its replication or again it'll go off into cell death. And of course at the end of G2, right before it goes into mitosis, it checks to see again if there's DNA damage. And if there's severe DNA damage, something that the system can pick up, then the cell stops. So these are the three checkpoints during the cell cycle where the cell checks to see that everything's going well, and if there's any issues, the cell does not go on to M phase, where it does cell division. And we're going to go through cell division quickly when we get back from our Thanksgiving break. So our key points are that the things you must know, and they're written here for you on this slide and the next, is that G1 is where the cell does a lot of its growing. It's in some cases, in some specialized cells that are not going to be doing any cell division, for instance, mature nerve cells, they go into G0 phase, where they just kind of sit in a dormant phase. Now that dormancy is called quiescence. Quiescence, and let me spell that out for you here. It's Quiescence. and it means dormant. Okay, it's the same as the word dormant, and it's important that you know that, because that's one of the reasons why cells arrest or go into G0. The other reason that a cell might go into G0, if it's badly damaged, if it's an aging cell, the cell would go into something called senescence, SENESCENT, senescent. And this is where we're talking about aging or damaged cells, right? And in this type of cell, in these older cells, okay, they can't do their functions very well. And because they're losing their ability to do their functions well, the system shuts them down. They end up going aging into cell death. So senescence in senescent cells never come out of the G0. They just continue in the G0 and then finally end up being reabsorbed into the body. Where quiescent cells do sometimes come back out of G0 and continue on in the cell cycle. So some tissues do this. Some just slowly go through G1. They grow bigger, they do get ready for DNA replication, but don't ever actually enter S phase, or they enter it rarely. And others do this cell cycle a lot. So in skin cells, you're going to have this happening a lot. In heart muscle cells, you might not have this happen a lot. In people that work out, their muscle cells are probably going through the cell cycle a lot more than in people that don't work out. Nerves probably go into, most of our nerve cells are in G0 phase when we're adults. Some might come back out of G0 phase. And as we get older, more and more of our cells go into G0 because of senescence. And so those are the kind of phases that's going on in a cell's life. And of course, a cell's life translates into the life of your tissues, which translates into the life of your organs, and then you as an organism. Here you have some pictures of cells going through the different phases. This is mitosis. We're not going to worry about that right now, but here's G1. Here's early S phase, middle S phase, and late S phase, and then G2. In S phase, you're replicating the DNA, which we're going through now. How we do this, that's what we're going through when we're talking about DNA replication. You saw the video I sent you earlier. You also did an activity today concerning DNA replication, and tomorrow we'll go over DNA replication in detail. Make sure you understand the important points. I'll send you an outline as well this evening. Then we'll talk about transcription and translation. Hopefully we'll get all that done tomorrow, and you'll have some activities to do over break, a virtual lab, and some questions to answer, and some videos to watch, a couple short videos like this one, 5-10 minutes. Take some notes, make sure you understand the key points, and when we get back, we'll go on into mitosis and meiosis, and we'll be able to hit genetics pretty hard when we get back in January. All right, I hope you understood all this. Again, I want to emphasize we're talking about a cell cycle here. Every cell goes through it. I want to emphasize the important points. G1 is where there's two phases, two major phases. M phase, which is the mitotic phase, the cell division, and then there's interphase. The cell spends 78% of its time, give or take, depending on the cell type, 78% of its time that's 78% of its lifespan in interphase, okay? 78% of its time. That's a lot. That's most of its life, not dividing. And then, of course, it goes into division when necessary, as it becomes obsolete or becomes damaged or becomes aged. It gets replaced, or it just ends up dying and doesn't get replaced. All right, so we're talking then about when we break interphase down, we're talking about G1, where it spends a lot of time growing, getting bigger. S phase, where it does copying of the DNA, which we'll talk about again tomorrow. You have a video on it. We'll talk about what this means. And then you go into G2, which is again more replication, more DNA, more replication of proteins that are necessary for mitosis. Here you did replication of proteins that, or making of proteins that were necessary for the replication of DNA. Here you do things. The cell gets bigger, duplicates its organelles. So if you have 40 mitochondria, you're probably going to want 80, because at the end here you're going to want to have 2 cells. You want each of them to have 40, so you double all your cell organelles. Here you doubled your DNA. Here you doubled all your organelles. Here you made all the proteins and collected all the resources necessary to do the mitotic division. Here you do the division, and you end up with 2 cells, each having half as much information, or rather having equal amounts of information, equal amounts of resources. And at the end, both of those then can go through the same cell cycle. So that, and this, ideally, this process is infinite. So G2 again is where you're taking and you're synthesizing the proteins that you need for the cell cycle. You're doing microtubules, so you're increasing the cytoskeleton. So you're getting ready for cell division, because if you're going to divide. Remember when we looked inside the cell, some of the videos we looked at, you saw that this is not just a sack of water. This has a whole cytoskeleton full of microtubules, whole roadways in every direction that allows a cell to completely control what's going on inside it. All that has to be doubled. So in G2, this is all that's happening. All these required structures are being doubled. In G0, remember what I said, you either get quiescence or you get senescence. And in senescence, you're talking about a cell that's dying. In quiescence, you're talking about a cell that's just dormant until it's necessary, until it's needed. And those triggers generally happen on the conditions in which the cell exists, right? So if the cell is signaled to replicate because there's damage, or if the signal is given to the cell to replicate because more of it is needed because of growing, because when you're a fetus, you grow into someone who's 6 foot tall, you're going to need more cells of every type. And then you have checkpoints that tell the cell, stop, there's something wrong, don't go into mitosis. And when that goes wrong, when these checkpoints go wrong, that's one of the ways you get cancer, because cancer is when there's a problem, a mutation in the DNA that there's nothing telling the cell to stop dividing. And so because nothing's telling this, the checkpoints are broken, because these checkpoints don't work, the cell keeps dividing and dividing and dividing. And this can happen in any cell type. So you can get cancer of the lung, you can get cancer of the breast, you can cancer of the testicles, you get cancer of the foot, cancer of the brain, bone cancer, blood cancer. All the different tissues of your body any one of those cells the checkpoints could go wrong, and the cell type starts to divide uncontrollably. So anyways, this is the cell cycle, and this is why the cell cycle is important. All right. Hopefully you watch this and you take good notes. Have a good one.